All right, well, I think we'll go ahead and get started again. Thank you all for joining. It's Mitchell Warren at AVAC, and delighted to welcome you to the next in our series of HIV Vaccine Awareness Day webinars. This one is particularly special because of all 5 in the series. This one has the most direct relationship to the HIV Vaccine Awareness Day history of 20 years ago, which you'll hear about in just a moment. But want to first just give you a bit of an introduction to the logistics for today, and then introduce our presenter, and then we'll go from there. Also to say that this series of 5 calls between the end of April and May 18th began last week with a webinar talking about one of the newer programs in vaccine development that is entering into later stage development from Janssen, the pharmaceutical company, along with a number of partners. That webinar recording and slides is available online, as will this one and future ones be. The easiest way to find that is if you go to avac.org/HVAD, you can see both the calls coming up to register as well as the recordings of the past. And we'll review that again before the end of this call. We are in a muted mode to keep the line clear, but I do certainly want to encourage everyone to engage in a conversation, and you can do that throughout the call by email to avac@avac.org, or you can post it into the chat feature on ReadyTalk if you're online. And then after Barney's presentation, we will also offer to open the line. People can unmute it, and I'll give instructions on how to do that. So that's kind of the quick order of the day. Want to especially acknowledge that today's call has a special opportunity because in addition to people calling in as they do for webinars, we have an amazing group of vaccine advocates gathered from all over the world, gathered here in Nairobi, Kenya, the Vaccine Advocacy Resource Group, and they're gathered together for this webinar, and so it's a great opportunity for real-time vaccine advocacy engaging with vaccine science. And just want to welcome the members of that phenomenal group, and you'll hear more about that throughout Vaccine Awareness Day and in a range of different approaches. As I said, this is the second in a series of 5, and want to just show the lineup. And again, if you go to avac.org/HVAD, you'll find this and you'll be able to click through. But you can see we talked about the AD26 program last week. Next week there are 2 webinars, one on the HVTN 702, a vaccine trial that is in an efficacy trial, next Monday. Then next Thursday, a week from today, an exciting conversation with Lynn Morris in South Africa. And I should say Monday's calls with Fatima Leher in South Africa. Thursday's also in South Africa with Lynn Morris, a leading neutralizing antibody researcher. And then it concludes the week after that on May 18th, the Vaccine Awareness Day itself, with an overview of vaccine development from Nelson Michael from the U.S. Military HIV Research Program. As I said, all of these calls are connected, of course, to HIV vaccines and HIV Vaccine Awareness Day. This year celebrating its 20th year, and you'll hear in the course of today's presentation about a very important moment in vaccine advocacy that culminated with high-level political leadership 20 years ago. And one of the clarion calls from then President Bill Clinton was to establish a center of great excellence in vaccine research at the U.S. National Institutes of Health. And 20 years later, as you will hear in a moment, if you look back on the 20 years of vaccine development and if you look into the future, the Vaccine Research Center, or VRC as you will hear, has really been at the forefront of a range of both vaccine and antibody-based activities. And we're delighted to have Barney Graham, who's the Deputy Director of the Vaccine Research, with us to give both a bit of the history of the center and what it's done, and perhaps even more excitingly, some of the future of what the Vaccine Research Center and its colleagues and collaborators are doing around the world. And Barney, I think you've been there from the very beginning, Barney, if I'm not mistaken. Is that a true statement?
Yes, I have.
He started in his youth at the Vaccine Research Center, now the Deputy Director, and has been a great educator, I think, for many of us in the advocacy world, both in some of the early vaccine development as well as in more recently the antibody world. And you will now hear that as I turn it over to Barney. Again, feel free to send questions as we go through the chat feature and at avac@avac.org, and we'll open up the line at the end of the call. But with that, I'll turn it over to you, Barney.
Thank you, Mitchell. and I hope everyone can hear me okay. Is it working?
Yes, it's loud and clear.
Okay. Well, this is a pleasure to be on the line with all of you and who are working so hard to still make this happen for HIV vaccines. And as I was putting this talk together, it's not only for me a history of the VRC, but it's sort of a personal history because I've been involved in HIV work since 1982 when I saw the first HIV AIDS patient in Nashville, Tennessee that had been diagnosed in the state of Tennessee. And then at Vanderbilt University, we were one of the founding groups of the, at that time, the HIV or AIDS clinical vaccine research program to test new HIV vaccine products. And so we've been at this a long time. And so what I'm going to do today is tell you a history of the Vaccine Research Center and how it came to be and how it's connected to this special day of HIV Vaccine Awareness Day. So next slide, please. NIAID is one of the 27 centers and institutes within the National Institutes of Health in the U.S. and it has an unusual status at NIH. It's the second largest institute in terms of funding, second only to the National Cancer Institute. But in addition to its mission to do basic research about microbiology and infectious diseases, as you see on the left, it also has a mandate to respond rapidly to emerging and re-emerging disease threats. And so, unlike many of the other institutes that continue to focus on basic research and translational science, NIAID has to also respond to unexpected outbreaks. Next slide. And so I'm going to go back to the mid-1990s. This was a time in which many things in our HIV world were changing. It was at the end of the era when we had really no antiretroviral treatment. So it was the beginning of the time when people could be treated effectively and live with HIV infection. It was a time when we had just discovered the HIV Coreceptor CCR5. So we started to understand neutralizing antibody a bit better. But it also was a time that was somewhat discouraging in HIV research because prior efforts had been focused largely on antibody. We learned that we didn't really know what neutralizing antibody meant when we found the coreceptor and found that laboratory isolates were not behaving the same way as primary viral isolates, and much of the work that had been done the previous years was based on a false premise. And so when that coreceptor was discovered, Dr. Tony Fauci, who's shown here in the middle at the board, was invited to the White House to talk to President Clinton and Vice President Gore and explain to them about HIV viral tropism. And I suppose part of this story is that unless you can get a buy-in at the very highest levels of leadership, it's hard to make things happen. But if you can accomplish that, many things can happen. So after this talk, Dr. Fauci was leaving the room, and after having explained, and to their credit, they seemed to understand how HIV could enter either through CXCR4 or CCR5 and have dual tropism. And President Clinton stopped Dr. Fauci as he was leaving and asked, what do we really need to solve this problem? And when you're with high-level political leadership, you also need to have an immediate answer at hand. And so Dr. Fauci immediately said, what we need is a center where we can bring multiple disciplines, scientific disciplines together to work in a center focused on finding an HIV vaccine. So next slide. So President Clinton turned to his chief of staff, Leon Panetta, who you know from other duties he's had since then, and said, make it happen. And so that was the beginning, next slide, of a process in which the Vaccine Research Center concept came into being. And May 18th of 1997 at Morgan State University commencement is when President Clinton gave a commencement address, and in that announced that he was going to establish a new AIDS Vaccine Research Center dedicated to finding a solution for HIV prevention. And so that's the reason May 18th is AIDS Vaccine Awareness Day, is because of the announcement of the building of this new center. Next slide. So since then, since the commencement on May 18th, there was a very rapid process, especially for the U.S. government, in which the building was designed in the fall of '97. Groundbreaking occurred in 1998, and June 9th of '99, a cornerstone was laid in a big ceremony, and at that time it was named after Senator Dale Bumpers and his wife Betty Bumpers, who had been big vaccine advocates in the U.S. for many years. They were from Arkansas, so were known to President Clinton. That's the reason this center is named the Dale and Betty Bumpers Vaccine Research Center. Next slide. And then it went on and was able to open in the year 2000 to start setting up laboratories, and the laboratories really were able to be functional in early 2001. And over that time, a group of basic research scientists combined with people who do clinical work and manufacturing have been assembled to work on the problem of HIV vaccine development. And I'll tell you a bit more about that. So the building is on the left. The current investigators are on the right. Some change and movement in and out has occurred, but we still have a relatively small number of investigators working on disciplines that range from very basic virology, structural biology, all the way through animal experiments, manufacturing, and clinical trials. Next slide. I reached back to some of the talks that I gave around that era of in 2000, and we all came here to the VRC with a specific mission to develop an HIV vaccine, and at that time all my talks were titled AIDS Vaccine Development, a Global Imperative. Next slide. The reason for this, and you may or may not remember these data, but at that time, even though antiretrovirals had just been introduced that were starting to be effective, still in the late 90s, we were having more than 5 million people a year infected with HIV. And it just felt like an overwhelming flood that we had to find a solution for. Next slide. And at that time, it was the second leading cause of death worldwide, leading to almost 3 million deaths each year from HIV. So 5.5 million new infections and about 3 million deaths a year from a single virus. Next slide. And it was recognized, and this is what happens when things become political. It does become political, but part of the motivation was that it was felt to be of economic and security concerns, and so this is part of what was driving development at that time. Next slide. So I'm just going to briefly review some of the vaccine approaches that have happened over these last many years, and then go into what we're doing now. And so I've listed the 4 major approaches that have gone into efficacy trial testing for HIV vaccine development. And the top bar are the subunit glycoproteins from envelope. The light shading is the early phase, and the darker shading is advanced development phase for each of those bars. Second bar is the poxvirus vectors, which would include the ALVAC study with a protein boost that went through a couple of phases, and so it has 3 different phases, but the later brown phase is where the efficacy trial was done in Thailand. The next one, the AD5 GAG-POL-NEF, the one without envelope protein in it, done by Merck, is shown in the 3rd bar with the blue shading. And then the Vaccine Research Center in its early days developed a DNA-primed AD5 boosted envelope GAG-POL-NEF DNA AD5 vaccine concept that was tested in the late 2000s. And so what I'm showing you on the bottom are how the concepts have changed over time. So antibody-focused protection and immunity is in the black line. Focus on CD8 T cells is in the red line. And as I told you in 1994 and 5, when the coreceptor was discovered, and it was discovered that primary isolate HIVs had a different biology than laboratory strains of HIV, antibody-focused work really lost favor. It was also found at that time a few broadly neutralizing antibodies, but they were relatively weak and not as broad as they needed to be. Those were antibodies like 2F5, for instance, and B12. So during that era of the late 1990s and 2000s, most of the focus was on how could CD8 T cells be used to rapidly clear virus and hopefully cause some abortive infections or at least control disease. And at the end of the Merck study, the AD5 study, the STEP trial in 2007, when we found the adverse result, interest in CD8 T cell immunity started going down. When the ALVAC study in Thailand, the RV144 study, had a result in 2009, the CD8 T cell response interest started going down even further because it was considered that most of that immunity was being mediated by antibody, although not completely true. There was some CD8 T cells involved. And then another basic science result designated by this gray bar was when a lot of new human-derived broadly neutralizing monoclonal antibodies started being discovered, like VRC01 and others that I'll tell you about in a minute, putting the focus largely back on antibody-mediated protection. Now, this is a mistake I think the field makes, is it has swings that are too extreme, and we clearly are going to need both antibody and CD8 T cells for complete and effective long-term immunity. But I'm just showing you the history here of how the focus on immunity has shifted. Next slide. And of course, I didn't mention it, but in the green shaded bars, the DNA AD5 trial failed and was shown to not be effective. It didn't cause any adverse effects, but it didn't work. And so that occurred in 2013. And so since then, the whole field has been really revising and restructuring what we're going to do. But we still need an HIV vaccine, even with all the ARVs that are so effective for people. There's still more than 5,000 infections happening every day globally, and about 5 people are infected for every 2 placed on ARVs, so we are not getting ahead of the curve. Even though the frequency of infection has been dropping somewhat, we're still getting more and more behind, in my opinion, and we do not have control of the epidemic, and still really need a vaccine. Next slide. So at the VRC, we don't just work on HIV anymore as we started out. We have expanded that work into a number of other areas of emerging infections and biodefense, and have worked on all the viruses and other diseases that you see listed here. One of the great privileges of being at the VRC and advantages we have is that we don't just have a basic research building shown in the upper left. We also have access to other components of vaccine development. So we have an engineering lab that can do process development and prepare for good manufacturing process, and that can be performed at our pilot plant in Frederick, just north of here. That can make GMP quality material from the concepts developed in Building 40 and get it in a vial, and then it can be evaluated in sync in very short order by the clinical trials program that is here on the campus at NIH in Bethesda, Maryland, shown in that next image. And then laboratory clinical samples can be evaluated and processed by robotics and other high-tech ways of analyzing immune responses at our immunology laboratory. We are somewhat dependent on partners, though, for advanced development. So this can get us through phase 1 testing and early phase evaluation of a vaccine, but to really expand into larger scale trials, we're very much dependent on networks like the HVTN and the HPTN. And we've done work with the US MHRP groups and with IAVI. And so we're very dependent on all of you and all the advocacy and clinical trial networks that have been set up to continue the work from here. And so what I'm going to tell you about now is how some of these other programs have helped us advance the cause for HIV vaccine development. Next slide. So in this process over the last 16 or 17 years, we've developed a number of platform technologies. So on the left you see an HIV virus, and from that we have developed ways of delivering vaccine antigens by gene delivery that could be done either as DNA plasmids or in viral vectors like a pox virus vector or an adenovirus vector depicted here. Or we can deliver vaccine antigens as proteins, like the virus-like particle shown in the middle that has been developed for chikungunya virus, or these nanoparticles just below that where self-assembling ferritin molecules can come together and display 8 trimers of the influenza hemagglutinin. Or, as shown at the bottom, subunit protein trimers like for respiratory syncytial virus that are now in clinical trials. Or on the right, you can see human monoclonal antibodies, as I mentioned, that have been so explosively developed over the last 7 or 8 years are being developed now for both HIV and Ebola. So there's a number of technologies and ways of delivering antigens or ways of delivering immunity that have occurred because of the focus and intensity of work done on HIV. Next slide. So HIV has driven a lot of this. In the process, we've also contributed to the history of DNA vaccines, which is briefly shown here. And the reason I'm showing this is because in that phase 2B efficacy trial done with the DNA advector, it was the first time DNA has advanced to an efficacy type study. And later we used that same technology to show that we could induce neutralizing antibodies to West Nile virus. And now that work has been leveraged to develop a DNA vaccine for Zika virus, and that vaccine is currently in phase 2 clinical trials and is probably going to go into a phase 2B later this summer. And so HIV vaccine development has driven these technologies, including DNA and other nucleic acid vaccine development. Next slide. It's also provided the tools and technology to solve other vaccine problems. So this is just the demonstration that using the ability to rapidly isolate monoclonal antibodies and solve structures, that technology allowed us to solve the pre-fusion structure of the RSV trimer for the F protein, which is a protein very much like GP160 on HIV. It's a class 1 fusion protein. Its overall structure is very similar to HIV, and by solving that structure and identifying it as a different surfaces than the post-fusion form of F, which had been traditionally used. Next slide. And then finding stabilizing mutations to preserve it in the pre-fusion structure, we were able to change antibody or neutralizing activity and immunogenicity from low levels to 70 to 80 times higher levels with this new molecule that's preserving epitopes and solving what we call conformational evasion of immunity for RSV. And because RSV doesn't have a lot of glycosylation or genetic diversity or immunodominance problems, this one thing may have solved the problem for RSV vaccine development, and this molecule now is in a clinical trial that just started in February that should help us know that by later this summer. Next slide. That work has also motivated the solution for a pre-fusion structure of the spike glycoprotein, another class 1 fusion protein depicted here for a beta coronavirus. So beta coronaviruses now have a solved trimeric fusion protein, a target of neutralizing antibody, and stabilizing mutations for this are working not only for this HKU1 beta coronavirus, but also for MERS and SARS coronaviruses. And so work on HIV and RSV now has maybe led to a solution for beta coronavirus vaccine development. Next slide. This, the interest in VRC, unfortunately, now at political levels, and just keep this in mind, that the only presidential visits we've had to the VRC have been for Ebola. And so even though HIV is still probably the most deadly virus on earth, still at a political level, at a media level, it sometimes doesn't get the attention it deserves, even though, in my opinion, it's still the king of all emerging infectious diseases. Because as Beatrice Hahn showed us, HIV is a zoonotic emerging disease that came out over the last several decades. And so I think in some ways it may help us to think of HIV as an emerging infectious disease and get it back higher on the political radar. Next slide. So work on Ebola and flu and coronaviruses and RSV, we're trying now to apply back to HIV. And in fact, these programs that are going on are informing our work on HIV because, as I said, these major mechanisms of immune evasion from neutralizing antibody, they're all active or prominent in HIV biology. This is what's made it very difficult to develop an HIV vaccine. But for these other viruses, only one or two of these features are active. And so by studying these problems one at a time in these other virus programs and vaccine programs, it is informing what we're doing with HIV. Next slide. And even though the core structure of GP120 was solved by Peter Kwong back in 1998, it's just been in the last couple of years that we have now the full pre-fusion trimer structure of the envelope protein for HIV. And that has allowed our group and other groups to stabilize this. So the work in Peter Kwong's lab here and John Mascola's lab here has led to stabilized trimers that are the core feature of our next set of immunogens that are being developed for HIV vaccines. Next slide. The work on antibodies has also led to some new understanding of how antibodies work and how antibodies are selected from our repertoires to develop over time to be effective antibodies at neutralizing viruses. And so one of the things that's been discovered here and other places around the world is that antibodies can recognize different epitopes, but sometimes there's a way an antibody can recognize a spot on a virus protein that is shared by multiple individuals. And so this would be an example of VRC01 or VRC01 class of antibodies that come out of the VH1202 heavy chain allele families. And so these antibodies, this is an overlapping 11 different antibodies binding almost exactly in the same way. And so by understanding how these antibodies work and where they come from, we now can even identify antibodies by sequence that end up being neutralized. So these antibodies have signatures that can be identified just by sequencing and understanding what elements are needed in the light chain and the heavy chain of an antibody to actually work. And this is informing our design of new HIV vaccines. It's also guiding some of the work that's being done now on influenza, which I think will also feed back again to HIV. Next slide. And so I'll tell you a bit about VRC01. It was one of the first broadly neutralizing antibodies discovered in that new era back in 2009 and 10, along with some others from other groups like PG9 and PG16. VRC01 binds the CD4 binding site, and it has breadth and potency that we think is sufficient to demonstrate some proof of principles. And so over the last 7 years and eight years, we've been pushing the development of this, and it's now it's been reviewed by consensus panels on multiple occasions, shown at the bottom. It's been manufactured. It's been tested in phase 1 trials in both HIV uninfected and in HIV infected people. And it has been tested in babies now as an adjunct approach to try to prevent mother-to-child transmission. And in about 1 year ago, in April of 2016, it was entered into a large efficacy trial that's going on in 3 continents called the AMP study. Next slide. The AMP study is to ask the question, can a neutralizing activity in human serum prevent HIV infection in people at high risk of infection? So it's antibody-mediated prevention study. This study is testing 2 different dose levels of antibody treatment given every 2 months. It's being done in North America, South America, and South Africa, either in high-risk men or high-risk women. And the goal is to enroll 4,200 subjects. We're up to a little over 1,700, almost 1,800 enrollments now. So the enrollment is being done by the HPTN and HVTN in collaboration, and it's going very well. It's probably being worked on by many of the people here on the phone. And so the enrollment is actually exceeding projections, and we hope that by the end of this calendar year, we could have this study enrolled, and then within a couple more years, be able to answer the question, if we can induce this type of neutralizing activity or this kind of functional response with a vaccine or deliver it with a monoclonal antibody, can we prevent HIV infection? Next slide. So this lineage-based vaccine design has also been coming from this work, and now we know that you can have even more potent antibodies than VRC01, and we know the characteristics of those antibodies, and so this work has provoked new ways of not only designing the antigen for the vaccine, but of delivering the antigen. And so this is a study that was done by Bart Haynes and reviewed in this paper by John Moscola, our director here at the VRC, and Bart Haynes, demonstrating the concept that if you could start an immune response that can induce the right kind of initial antibody, even if it's not neutralizing at that point, can you push it into the evolution it requires to become a broadly neutralizing antibody by sequential immunization with these antigens shown on the left to push that antibody development into the right direction? This kind of work has been done, and you'll hear more about this probably from Lynn Morris, where the CAP256 antibody has been discovered in an analysis like this from South Africa. And it's one of the monoclonal antibodies that we're working with in collaboration with them to develop as a passive therapeutic option for testing later. Next slide. So in summary, this is my last slide. The NIAID Vaccine Research Center is connected to the history of the AIDS Vaccine Awareness Day in the ways that I showed. Its building was announced on May 18th of 1997. We're still very focused on developing vaccines or monoclonal antibodies that can prevent HIV infection. All of us came here, and all of us are still devoted to finding a solution for HIV. And everything we do on other products or other pathogens is we try to apply that back to HIV. And but in the meantime, we're also advancing some of these vaccine concepts that are relevant to other viral diseases. So thank you very much, and I'm glad to take questions.
Fantastic. Barney, thank you so very much. That was a terrific overview, both of the science, but also of the history, and as you started with the importance of political will and advocacy. So just a great combination for the webinar. Thank you so much. As I said at the outset, you can continue, and many people already have, put questions in the chat feature on the ReadyTalk platform. You can continue to email at avac@avac.org, and would love live questions as well. You can hit *7 to unmute your phone, and then I would ask *6 to remute so we keep the line clear as we are recording it to be able to post it on the web for those who aren't able to join this webinar live. So perhaps I have a bunch in the queue, but perhaps I will pause just for a moment to see if anyone would like to jump on the line with *7 to unmute your line, and again, introduce yourself if you would. While people are formulating their questions, let me just jump in with one that I think is really an interesting one, because you described, Barney, that a lot of the history of some of the early vaccine candidates that the VRC developed and took into clinical testing, and then moving on to the VRC01. And a question came in, what plans for, what might come next? Are there products, obviously further behind VRC01, that you are advancing currently in phase 1 that you think might be promising getting into efficacy trials in terms of what candidate and what timeline might you see?
Well, there's programs for both vaccine antigens and for broadly neutralizing antibodies. And our focus is on prevention of HIV infection, but we're also exploring whether antibodies can be used to treat or be used in the process of curing HIV infection. So VRC01 is the first in a series of antibodies that are being tested. Like I say, one is CAP256 that we're doing in collaboration with Lynn Morris and Slim Karim and others in South Africa. We're trying to look at antibodies to different regions or different epitopes on the envelope. And so There's a program on an antibody called 10E8 that hits the Emperor region. We're working with Dan Baruch and others on PG121 to see if we can use that. We're working on other antibodies that can hit the V2 region, and other more broad and antibodies that can hit the CD4 binding site, and also adding extended half-life mutations to these antibodies, because there's ways now, and if you had a more potent antibody, and there's ways now of extending the half-life so that you could potentially do treatments subcutaneously every 3 to 6 months and maintain a high level of immunity. So there's a lot of work like that going on with the antibodies. The antigen design process has come back around, and there's 2 major approaches now to induce neutralizing antibodies from HIV antigens, either to the fusion peptide or to the entire HIV envelope, and it's all based on structure design. Most of this work is coming out of Peter Kwong and John Moscola's lab programs. It includes approaches to deal with the glycans that cover up a lot of these epitopes and hide them. And it's interacting with our flu program to look for ways to overcome genetic diversity. So those programs, the antigens, are probably not going to be back into phase 1 clinical trials until 2018 or so. But the antibodies are going to come out in a succession of trials. And so we're hoping to have some results from this AMP study within a couple of years, and by that time there should be combinations of antibodies that would be ready to go into other high-level advanced efficacy trials that could be even more effective and more practical.
Great. Thanks, Barney. One of the questions came in. Oh, great. Is there a question on the line?
Yes. Hey, everybody. Hey, Barney Graham and Mitchell. This is Matthew Rose with the Bard. And one of the questions that we had floated around in our discussions are what are we looking at in terms of being able to elicit these antibodies in individuals, knowing that giving an infusion will not necessarily be a practical use in the terms of real-world application? Right.
So right now this AMP study is being done intravenously, because even though the VRC01 is broad and potent, to get enough to really be effective and test the concept, it's being given intravenously, and that's not a long-term solution. So these more potent antibodies that are sometimes 10 to 100-fold more potent and are potent enough and can be made for subcutaneous delivery, and with the long half-life mutations could be given subcutaneously, for instance, every 3 to 6 months, which, you know, is not. Many diabetics do subcutaneous injections every day, and so you could envision that as being practical. But we're also trying to design antigens to elicit these kinds of antibodies. That is a much more difficult task. If you can remember that slide I showed of the conformational evasion and glycosylation and genetic diversity and immunodominance problems of the envelope, all extreme cases of the HIV virus defending itself against neutralizing antibodies. But we are designing antigens, and we feel like eventually, because of the atomic level information we now have of the details of the antigen structure, we will eventually get to a place where we can make these kinds of vaccines that can induce the right antibodies.
Great. Well, thank you, Matthew and Barney. A question came in related to a slide you presented early on about some of the history and that the oscillation and the pendulum swing between antibody and T cell. And a great question from someone asking, what would it take for the field to move to a longer term view that doesn't oscillate so drastically with each result?
Well, it takes a little bit of scientific diplomacy, maybe, and honestly, the other thing it takes is more money. And part of the reason for the extreme focus, either in politics or in science, is that there's not enough resources to have the freedom to explore things that are not in vogue. And so, unfortunately, this pendulum swinging is in part a consequence of not having sufficient funding to explore all options. So part of the advocacy here is got to be at the political level as well as at just the patient care level, which is obviously the most important. But there's got to be some advocacy at the political level because, like you say, in the long view, the important thing is, in 20 or 30 years from now, will we be in a position with the right knowledge base to not only deal with HIV, but to use that knowledge and be able to deal with every other emerging infection that's going to be coming because of the way we're affecting our environment.
Barney, a series of questions came in on a somewhat related topic because you described experience within the VRC even of presidential visits because of Ebola and obviously work that you all are doing there and with Zika, even as you maintain a prioritization with HIV. And a series of questions about, are there lessons from that we could take? What were the drivers of that, both in terms of the politics and funding, and then a related question about some of the scientific drivers that got us so quickly, or seemingly so quickly, to an Ebola vaccine that was shown to be safe and effective relative to HIV. So I guess both the question of scientific and political drivers, and are there lessons for us?
Well, even at the political level, most things are personal. And so things that become personal are more effective. And so, for instance, I'll never forget in the mid-'80s when finally Ronald Reagan was able to provide additional funding for HIV research, but it was in our opinion, it was largely motivated by the death of his friend Rock Hudson. So it was personal. In the 90s, when ACT UP was so active and getting all the media attention, that's really what accelerated much of the access to antiretrovirals. And it really wasn't until the early 2000s when there was demonstrations in the street and it became a media event in South Africa that the PEPFAR funding finally really came through. And so in my view, looking back, it's either got to be personal or it's got to have a lot of media attention that makes it personal for politicians. And if it's not personal, it's just not going to be effective. And so, somehow, if HIV can be recast as an emerging infectious disease, the most difficult one that we've ever faced, and if we can find the solutions for HIV, then that will protect us from all the other things that are coming in the future. And then put the attention back on HIV to solve this problem once and for all. And as a byproduct, then your families will be safe from pandemic influenza, from Zika, from all these other things that are going to keep coming. Now, the problem, I think, we had been working on Ebola. Nancy Sullivan and Gary Nabel had been working on Ebola even before the VRC started. And so some of these things become easier because of the biology of the virus, not because of the effort put into it. And HIV just happens to be an extremely difficult biology, and that's why solving HIV, I think, will solve all the other problems and future problems we're going to have with viruses.
That's a great point. I think you had a number of examples in real time in your slides of where both HIV has influenced and helped other areas and vice versa, and I think making that point both scientifically and for advocacy is so important. Another question came in. You mentioned in a number of places the broad numbers of collaborators the VRC has. So obviously you're a center working within the NIH, but you mentioned a veritable who's who of organizations that with which you partner. And a question, given resources, which are clearly limited, what role for the VRC itself, but also just your sense of the field right now in 2017 in terms of how to further leverage and encourage the pooling of resources and capacity, particularly thinking about screening and prioritizing future candidates, because clearly not all concepts can go into clinical efficacy trials, even if they all pass go, no-go criteria, just because of resource constraints. And just some questions about how will the VRC, but importantly, how will the VRC within the ecosystem of the field prioritize some of these decisions from your perspective?
Yeah, well, as a field develops, some of you have probably noticed this too, but as a field develops, sometimes it loses track of the original mission or the original need, and maintaining the organization becomes as important or more important than solving the original problem, which would hopefully put us all out of business. And so, I think one of the things that's happened over time as the HIV research field has grown and expanded is that sometimes we focus more on maintaining our organizations than on solving the problem. And so what happens is we get siloed into different groups or factions that all want to see that particular thing go all the way through. And there's really no election or adjudicating body that makes these final decisions of how things go forward. It's not like a global company that has a hierarchy and a decision-making process. And so these decisions about what actually goes all the way forward and gets into the efficacy testing is largely determined by the people paying for it. So it's determined by the funders. And so, the philosophy at the VRC is that we're just going to focus on the biology and focus on the science that makes sense to us, and then, hope that will win arguments at the end if it is better. But, honestly, our philosophy here, because it's a public institution and we don't really have to focus on commercialization or profit, is that we just want to see a solution. So whether it's our product or someone else's product, if it has the best biology and if it has the best scientific merit and a scientific hypothesis, then we're all for testing that. I do think it's important to try to test things that have a distinctly different hypothesis for how they might work. So that we aren't just repeating the same experiment over and over, that we really try to explore new concepts and advance things that are not just incremental, but can be more of a fundamental change.
That's great. Thank you, Barney. Let me pause. I have a few more in the queue here, but let me pause to see if anyone else on the line wants to unmute with *7 and ask a question before we get closer to the end. So one question came in, Barney, and it's funny. I think many of us who have watched this field, you said in a great presentation there was one thing. You mentioned the HVTN 505, the trial of the AD5 from the VRC, and you used the word failure, and I think many of us would say, trials don't fail even if they don't have the results we hoped for. And not to correct you there, but wondered a question from someone on the line. As you look back now, having been very involved in that program, what lessons do you derive from 505? And in your mind, was it a failed trial, or was there something gleaned from that disappointing result that still helps you think about answering a question from that trial?
Well, I probably used the word failure because it was such a great disappointment to me personally because of all the effort and time we had put into getting to that point. But I think the lesson we learned from that is that we have to know more. We have to know more about the details. I think ultimately the reason that trial did not work is that maybe we didn't, in retrospect, put enough attention into the real atomic level understanding of the structure of the antigen. The antigens were not designed well enough for antibody responses that we needed. At the time, we didn't understand the biology well enough to design them well enough. So, I don't know if we could have done that much better even at the time. But the concepts that we advanced were I think it was important to carry it through and get an answer, and the answer was negative. And really what it taught us is we have to know more. We need to know more details about antigen structure. We need to know more details about mechanisms or location of T cells, not just to have a T cell response, but to have a T cell response with the right function and the right location. And so it really taught us that we need to know more. One of the problems that many of you have probably noticed with vaccines is that it does take a long time to go through the development process. And so most of the things that we're testing are concepts that are anywhere from 5 to 10 years old. And so a lot of times when we're testing a vaccine, it feels like we're using old ideas and old technologies, and the science is moving so fast that one of the goals here in some of the Zika and other programs we've done is can we make the manufacturing development process faster so that we're testing concepts more in real time. And so part of what we're doing here is trying to we've been in a hurry since we started, but we really want to have the manufacturing development process faster so that we can be testing real-time immunology concepts and knowledge of the viruses as close to real-time as possible.
Great answer, and I think a real sense of what one does and learns in a trial, irrespective of the primary question being answered. Before we wrap up, there's one great question that I just thought really perhaps is the best way to conclude, and that is, you've done both an incredible scientific primer, but you've also actually, as a scientist, helped advocates understand some of the history and the advocacy needs. And a question from a great advocate saying, as you look going forward, and particularly given the current politics and funding threats generally, are there low-hanging fruits, so to speak, that global vaccine advocates should aim for to support the current and future work within the VRC itself and within the HIV vaccine community writ large?
Well, I don't know if it's low-hanging fruit, but, I've always thought that people could understand a lot more than we give them credit for. And so I've always thought that if people understood how vaccines work, everybody would want a vaccine, everyone would want to be part of vaccine programs. And that people, even lay people, if you get the language right, can understand immunology and virology concepts, because honestly, they're not that complicated. It's really a matter of communication and language. It's the concepts themselves are not all that complicated. And so, to me, the thing that I don't think our politicians and many people may not realize, but what a revolutionary change there has been in vaccine development capacity just in the last 7 or 8 years. The advent of these technologies for rapid human monoclonal antibody isolation, high-level, high-throughput sequencing to define the variability in viruses and the details and the variability in our immune responses at the level of repertoire analysis, the rapid ability to define structures of proteins, the flow cytometry capacity that's giving us analysis of single cells so we can do epitope-specific phenotyping of cells, and it's These technologies have completely changed the way vaccines are conceived of and are done. And I think to have fiscal constraints in a time when there's so much opportunity to make advances, not just for HIV, but for all infectious diseases and vaccines to manage a lot of other problems, that I think education about the technology revolution that's going on that most people may not recognize might be the best way to spend time.
Barney, that is terrific. On so many levels, including the importance of science and research literacy, not just for HIV vaccines, but for so many elements, is a great reminder. And I think the other thing implicitly that you just described is as a leading scientist at the Vaccine Research Center, you are yourself a great advocate, and advocates can also become great citizen scientists, and just really want to acknowledge that and thank you for that. And also want to let everybody know that this was just number 2 of 5. As I said at the outset, but as you'll see on the screen, this conversation continues, and it doesn't just continue for 2 more weeks to May 18th. It's a conversation that we hope continues always around HIV vaccines. And again, last week about Janssen, and now Barney's presentation 2 will join it on the website at avac.org/HVAD. But certainly encourage you to also go there to register for the next 3 conversations on building on the status of the HVTN 702 trial, building on the RV144 results from Thailand that Fatima Lahir from Pakistan. Next from today, entitled Plan B NAB, an overview of antibody research from Lynn Morris. You got a taste of that from Barney today, and we're delighted that Lynn will join us from South Africa next week. And then the week after that, Vaccine Awareness itself, the so-called vaccine hunter, Nelson Michael, from the Military Research Program, is going to do an overview. And I think that's a little bit of thoughts even that you heard from Barney today about HIV vaccine development in the context of other vaccine development. And really hope that you'll join us then for those conversations. Want to thank everybody for joining. I realize we've gone a little bit over time, but totally worth it. And I'm delighted that everybody, by my look on the Ready Talk, is still online and engaged. So thank you so much for sticking with us. And Barney, thank you so much for the science lesson, the advocacy, history lesson. And looking forward to many more opportunities.
Okay. Well, thank you all for all the help you've been to us.
Fantastic. Well, everyone have great evenings, afternoons, or the rest of your mornings, depending on where you are, and we look forward to talking to you on Monday. All the best.